I found this fascinating that the rise of Nazism and eugenics had a serious effect on quantitative genetics. And then, right in the beginning of our conversation, you also mentioned that there were controversies in the 1960s and 70s. So I saw that there was a paper by a psychologist named Arthur Jensen. I think the title was "How Much Can We Boost IQ and Scholastic Achievement" or something like that, and it came out in '69. And these two things together, I mean, they're emblematic of the controversies that quantitative genetics faced over this 100-year period. And I thought that before we get into the findings, we might just talk about what these controversies were and whether or not they had any merit.
Great. Yeah, that's good. That kind of finishes the story we began earlier about these two worlds of genetics, molecular genetics and quantitative genetics. And I explained why they diverged for most of the century, because they just had very different goals. Molecular genetics trying to understand how genes work, focusing on single gene disorders, creating mutations, like big sledgehammer effects to understand how the genes work. Whereas the quantitative geneticists, following up from Galton, wanted to study traits, and they wanted to use genetics to help them understand these complex phenotypic traits. So then, because quantitative geneticists thought there were many, many genes involved, they didn't pay much attention to molecular genetics because they knew these single gene approaches weren't working. But what happened in 2003 was the human genome sequence, after 10 years of $2 billion worth of research, sequenced the whole human genome. And then what followed from that was understanding that there isn't a human genome sequence. We all have unique DNA sequences. We differ with millions of these DNA base pairs. So there's human genome sequences. And then once you find these DNA differences, you can ask, do these DNA differences relate to complex traits like behavior? So in '85, these two worlds started coming together when we got the first DNA markers of DNA itself. So for 50 years, we've had DNA markers like blood types, but they're not DNA differences, they're phenotypes. Blood is a single, blood types are single gene phenotypes. But by 1985, as we could begin sequencing DNA in 1970, so that's when this started. And then by 1985, there was a technique developed by Alex Jeffreys in Leicester in the UK called DNA fingerprinting. It involved a type of DNA difference that is highly variable between people. It has to do with the number of repeats. And it gives you like a barcode then across the genome. And so that's what's completely transformed forensics now, because you can uniquely identify a person. You can say, the very famous case in the 1980s in England was there two adolescent girls had been raped and murdered, and they didn't know who it was, but they had sperm, and so they knew they had the DNA. So they decided they would take this. This is 1987, so this technique was only first developed in 1985. But it was so powerful that they thought, well, they could just get DNA from all the men in the area and determine who the killer was. So, you can imagine if you were Sir Alex Jeffrey, you would be quite worried about this because, it's a new technique. Is it really going to work that well? So they rounded up everybody, every male, and there was pressure on them, to provide a DNA sample. They didn't have to provide sperm. They just have to provide a little cheek swab of DNA. So they got everybody, no one. And then, so they thought, oh God, here's another crazy science experiment. But then some woman heard a guy overheard two guys talking in a bar, and the guy was saying, I'm not going to do that, and this guy had forced him to give a DNA sample for him. And having heard that conversation, they got this guy, got his DNA. It was him. And that he had this Dickensian name, Pitchfork. And he's been in jail since, and they just released him last month, which created a bit of a furore as well. And this forensic thing has been very interesting because it's been responsible for getting something like 30 people off of death row. Every now and then you hear about one of these cases where in England, just last week, there was a guy or last month, who had been in jail for 17 years for a crime he never admitted, a murder he said he didn't commit. And it turns out it's a real scandal that there was DNA, but the police said there wasn't DNA. And the DNA proved it wasn't his DNA, so he was let out of jail after 17 years in jail. So people know about how it's used to identify criminals, especially through these large DNA databases now. But I think it's very interesting to see that several dozen people have been let off of death row because the DNA proves that, the police were saying, this is their DNA, this is the blood of the killer or whatever, and it's not his DNA. So that was kind of neat to have that happen in the 1980s. And then as the human genome sequence was determined from the 1990s to 2003, that led to all sorts of unintended discoveries. I think it's such a great advertisement for blue sky science because, everybody thought pretty good idea to sequence the whole human genome. It wasn't so easy to say why, but it turns out all of the big discoveries that have come from it were unanticipated. And for me, it was finding these millions of DNA variants that you can genotype. And in the 1990s and 2000s, you had to do it very laboriously and expensively. One by one, you had to genotype people, get a little bit of DNA from them and genotype them. It's very expensive. But then the final advance was technological, something called a gene chip or SNP chip. It's a little array, DNA array, the size of a postage stamp that can genotype hundreds of thousands of DNA differences at a time with just minute bits of DNA. It's really an amazing advance that led to a Nobel Prize. And that's what's changed everything because it's made it very cheap. So 27 million people have paid to have their DNA genotype using these called the SNP chips because it's a particular type of DNA difference, the most common one, a single nucleotide polymorphism, SNP, SNP. And these chips then measure hundreds of thousands of these SNPs across the genome. So they allow you to sample variation across the entire genome. And they now cost, you could do this for about $40. And if you did use 23andMe or one of those companies, they'd charge you about $100 to do this, to get your DNA tested. So that then allows us to move from single genes, because that was the game in molecular genetics up until 2000 or so. Find a single gene disorder and be the first one to find the gene for that disorder. And if you get your publication in an hour before someone else, then you're the discoverer of that gene. And so and then you started running out of genes, and it was almost like clerical work. There was no intelligence involved. You just screen for all these genes and you find where it is, and then you do more intense studies to prove it's actually this base pair of DNA in the 3 billion base pairs that's responsible for that. So molecular geneticists, once the chips came out, started saying, wait a minute, now we don't have to just be stuck with single gene disorders. We can look for complex traits. And at the same time, quantitative geneticists said, wow, we can use DNA techniques to look for genetic variation. And so that's when it really came together. And it was a fusion that really led to a huge explosion of research that continues today. You can't keep up with all the advances.